Welcome everyone, and thank you for coming to the annual Picower lecture. And it is with tremendous pleasure that I introduce Carol Barnes today as our speaker. So Carol is the Regent Professor of Psychology and Neurology at the University of Arizona in Tucson, and she's also the director of the Evelyn McKnight Brain Institute. So for over 3 decades, Carol has made seminal contributions to our understanding of the circuit and molecular mechanisms to spatial learning and associated neuroplasticity. Her work illustrates how the structure, plasticity, and function of the hippocampus changes during biological aging. She has delineated the effect of aging on the dynamics of information processing. She has also demonstrated how expression of certain activity-regulated genes is altered during aging and in cognitive impairment. And I think that her body of work really helps us to understand how the cognitive function is compromised during aging and in human neurodegenerative disorders such as Alzheimer's disease. So Carol's work is well recognized, and she's the recipient of numerous awards and honors. For instance, she is elected the Galileo Fellow at the University of Arizona, and she also received the Micah Salpeter Lifetime Achievement Award from the Society for Neuroscience. She's a foreign member of the Royal Norwegian Society of Sciences and Letters, and she was the president of the Society for Neuroscience, just to name a few. And she's currently the chair of the Gruber Foundation Neuroscience Prize Advisor Board, and that was actually where I had the fortune to get to know Carol well. And it is really a tremendous honor for her to be our speaker today, and the title of her lecture is Normal Brain Aging, Impact on Circuits Critical for Memory Across Species. So please let us welcome Carol Barnes.
Thanks so much, Li Wei. I've had such a great day today, seeing old colleagues and new colleagues, meeting new colleagues. And this is specifically for Matt and for probably Ann Smith as well, who both lived in Tucson for a while. But this is actually out my front door, which they can attest to. So the main focus of the talk that I'm going to give today is on how circuits important for memory and the behavioral expression of experience are altered across the lifespan. And why I think this is important is because I want to be able to describe what happens in the absence of dementing illness as a backdrop for understanding disease processes that can be superimposed on normative age-related changes. I think there are chairs around there, and I really don't mind if you walk past and seat yourself down rather than on the floor. Okay. You're also poised for a fast exit, so that could also be important. So I'll give you an overview of what I want to do today, and I might sneak a couple more things in at the end that I'll tell you about. But first, what I want to talk about is the cognitive changes that we have observed on tasks that depend on the hippocampus. We know that the hippocampus is a structure vulnerable to normative aging, and we have lots of cognitive proof that hippocampal-dependent tasks show changes during old aging. And most of the data I'm going to show you actually is from the hippocampus, but I'm also going to show you describe changes in recognition memory that depend on the perirhinal cortex, and just the changes in working memory that rely on frontal cortical function. Then I'm going to review some data on basic electrophysiological changes that are known to occur in the hippocampus of old memory-impaired rats, including hippocampal responsiveness, connectivity, and plasticity mechanisms. And after that, I'll describe examples of how the dynamics of networks of hippocampal cells change during aging in the rat, and some implications that these results have for specific hippocampal subregions. And also I'll mention an experiment using a method that monitors behaviorally driven activity in circuits across the brain with single cell resolution, the catfish method. And with this technique, we've been able to assess changes in functional activity patterns over wider areas of the brain. And I'll focus this on the areas of the brain that we've examined in rats that I can compare then to areas of the brain that we've observed in aging monkeys that also go through these changes. Then what I'd like to do is end with a brief description of normative aging changes in perirhinal cortex and frontal cortical circuits, some relatively new data, and I'll show published and unpublished imaging findings in young and old monkeys and rats. So I think I can get through all of this, but I want to, at the end, I'd really like to show you one more thing, if there's any time. I want to say a couple things about progress we've made towards telemetered recordings of monkeys who are completely free to behave in large environments. And I can show you, hopefully, just a couple slides that will whet your appetite. And maybe 5 years from now, when I'm actually done with this study, you can invite me back and I can tell you more about it. Okay. So just to remind you of the kinds of tests that have been used to show hippocampus-dependent memory impairments across age and species, these are tests that involve navigation. They don't require language, and so they can be used broadly across species. For example, spatial memory deficits have been observed in old rats and old mice using the circular platform spatial memory test, and the Morris water maze. Rusty Gage first showed that older rats were deficient in the water maze. As you see here, circular platform task also relies on the ability of animals to use distal visual cues in the room to guide their behavior. And in older animals, again, there are spatial memory deficits day to day in this task. It's not only rodents that show spatial memory problems, it's also primates, including rhesus macaques. Peter Rapp's lab is consistent, has acquired data that's consistent with spatial memory problems in older monkeys. So there's a big platform you see here, and around the platform are locations where the monkey can retrieve favored food objects. And under these conditions, the older monkeys forget where they've already obtained the food and they go back to the wrong spot. And so they do show impaired spatial memory. I could show you many experiments in younger and older humans, but I pick the Newman and Kazniak study because it's essentially a dry version of the Morris water maze. And that is the experimenter and participant goes into the room, sees where the pole is, sees all the cues around the room, and then they go out of the room with the pole, and the participant has to put the pole right back in the right spot. And what happens is that older adults aren't as accurate at finding it consistent with spatial memory deficits in older people. And I think when I was talking to graduate students and postdocs this afternoon, I mentioned that what I'm really fascinated by is that spatial memory problems implicating hippocampal dysfunction exist at whatever age a given species is considered to be old. Okay, so that's 65 years in humans. I'm afraid I'm crossing the threshold this year. So I'm going to be old. Not yet. I'm going to be old this year, however. It's 22 years in monkeys, so you multiply by 3 rhesus macaques, and that's equivalent to 66 human years. And that's 2 years in rats, which is the 50% mortality in that animal. So a number of factors may contribute to these observed memory changes at the edge of the lifespan of any of these species. And I'll start with what was thought to happen to neurons during the aging process when I became interested in aging in the early 1970s. Okay, so these are entorhinal cortical neurons of a person who's 20, which is many of you in the room, and then over to somebody who is 90 years of age, where the neurons would be incredibly shriveled. And this is a particularly disturbing picture of aging brain because, as you know, your neurons are born largely before your birth, and they die with you at the end of your life. And although this picture still shows up in textbooks today, we now know that this depiction of brain aging is completely wrong. In fact, today, we know that if you just take hippocampal neurons, for example, cells in granule cells or C3, CA1 pyramidal cells, and you count them, when you count them stereologically across age in rats, mice, dogs, monkeys, and humans, these cells do not decline with age in any of these species. So one thing we can be confident that doesn't contribute to hippocampus memory impairment with age is global cell loss. Okay. And there's some more good news. Before I get to the not so good news, I'll give you some more good news, and that is for those of us who are determined to age successfully, many cell functional properties are very well preserved in the aged brain. When the activity of cells are recorded in the hippocampus in vitro, where hippocampal tissue can be kept alive with oxygen and artificial cerebral spinal fluid for a day or two, we and others have found that most biophysical properties or electrical properties of old hippocampal neurons are really quite intact. The old cells are healthy, and as I list here for you, most of their membrane properties are similar to those of younger animals. Of course, there's a couple exceptions. But if you didn't know the age of the cell that you had impaled and were recording from, you would be hard-pressed to tell me which was the old cell and which was the young cell. So you might say, well, okay, can we all go home? There's nothing wrong with the aged brain. Well, what is normal aging? If in the hippocampus there's no cell loss, most of the cells are healthy, why do we observe memory changes? Well, there obviously are multiple answers to that question, and one is that the change with age occurs in the connections between cells. There are fewer actual or functional synaptic contacts. So while hippocampal cellular preservation during aging is more the rule, there are frank anatomical and electrophysiological changes that can be measured. And I'm going to give you two examples, one for granule cells and example for CA1 pyramidal cells. Yuri Geinesman many years ago did the tedious, really beautiful, elegant serial EM, stereological reconstruction, and we know that there are fewer synaptic contacts from the medial entorhinal cortex onto granule cells. Electrophysiological data from my lab suggests that there is actual axon pruning of this entorhinal cortical projection in the old rats. But the individual synapses that remain in these older animals are actually more powerful, as though there was a compensatory process occurring. So this change means that fewer synapses can result in granule cell discharge. For CA1, very closely related structure, Yuri Geinesman also did the stereological reconstruction. There's no loss of synapses from the Schaffer collateral input from CA3 onto CA1. However, what's interesting, more recently, they found that a subset of synapses that exist in the old CA1, on the old CA1 cells, have this reduced postsynaptic density size. And the physiology suggests also no axon pruning, but rather a substantial loss of functional synapses along the dendrites of a given CA1 pyramidal cell. The unitary EPSP size at this synaptic contact does not change with age as it does for the granule cells. And this observation is at least consistent with the hypothesis that perhaps those synapses with this reduced postsynaptic density in old rats are actually silent. So what did I want to tell you from these examples? Essentially, it should be clear that synaptic connectivity is altered with aging. First, the numbers of physical synapses can change, and even if the number of synapses are the same in a particular region, the function can be dramatically altered. So, another thing that I'd like you to notice from these data is the incredible region selectivity and specificity of the changes that do occur in the aging brain. So even different cell types within a major structure can show different kinds of changes. All right, so another category, whoops, I'm not supposed to touch this. Okay, another category of change is plasticity at the aging synapse. And of course, it's now believed that the biological basis of memory can be found in the mechanisms that permit modification of synaptic strength in the connections made between cells. And what a mechanism, something like a mechanism like long-term potentiation, LTD, used by the brain, it can be used to store experiences. And both long-term potentiation and long-term depression, or LTD, change in hippocampal circuits with age. So under some conditions, LTP can be just as effectively induced in old rats. As you see in these schematic from old data here, but the decay of the modification of the synaptic strength is about twice as fast in the older animals as in the young animals. So the decay time constants here are 40 days for the young and 20 days for the older animals. Furthermore, this maximizes LTP induction, okay, these stimulation parameters. But if you use less robust stimulation parameters in old rats, these kinds of, you can see powerful LTP induction deficits as well. So these kinds of parameters do matter in the aged brain. Additionally, I want to emphasize that it's not only LTP that is altered, but LTD and LTP reversal are altered in the aged brain. Tom Foster's lab has shown this, that both of these mechanisms reduce synaptic strength. So clearly the plasticity mechanisms thought to be used for information storage in the nervous system are compromised in old rats, and it appears to be correlated with individual differences in learning. So when you relate the durability of LTP to the ability to remember where you are on this circular platform task, there's a strong correlation. So it is related to individual differences in memory performance. Okay, so from the observations of altered plasticity and reduced connectivity in the older brain, one would predict that there should be an impact on circuit characteristics and behavior-driven networks in freely behaving animals. What's remarkable to me is that it's actually 40 years now, over 40 years, since John O'Keefe and his student Jonathan Jastrzewski published the observation that pyramidal cells in the hippocampus discharge spikes in discrete and reliable places when they wander through space. John referred to the area over which the place cell fires as that cell's place field. And first, I'm just going to talk about younger animals. I'm just showing you a few of the cells that can be recorded on this triangular track. The different colors are different cells, and the dots represent the spikes. And these two panels illustrate place cell recording from a young rat in 2 sessions on a day, morning and afternoon. And you see that the same hippocampal map is retrieved on the second session as was there on the first. So the yellow still fires here, the red cell still fires here, and so forth. So the young rats retrieve the same map from session to session. And most of the time, the older rats also show accurate map retrieval. But on about 1/3 of the time for any given old rat, a completely different place cell field distribution appears when the old rat goes back into the same very familiar, well-learned environment. And you can see that here, this blue cell now fires here. The orange cell didn't fire in the first session at all. It's as though the old rat had retrieved the wrong map. So the observation that a completely different neural activity pattern can emerge in the presence of the same, physical well-learned stimuli could partly explain the consistent finding of poorer spatial memory in all species that have been studied. So for CA1 then, when the environments are familiar and identical, cell ensembles in old rats can occasionally remap when they shouldn't. A whole different thing happens in CA3. So I'm going to take you through CA3 now. So for CA3, Ian Wilson and his colleagues, using a different experimental procedure, showed that the opposite can occur in this closely related region. So when the environment is changed from one session to another, this is a circular environment, a square, and back to the circle, young rats show global remapping. That is, the pattern of cell firing is different in the square than in the circle. For old rats, on some occasions, they do not remap when they should and appear to reuse an inappropriate old map clear throughout the session. And this is clearly a different kind of retrieval error than you see for CA1. So old CA1 pyramidal cells remap when they shouldn't, and old CA3 cells don't remap when they should, suggesting that the dynamic processes of pattern separation and pattern completion are altered in hippocampal networks. So interestingly, findings similar to the ones that I've just described for Ian's CA3 data have been reported in the human literature, where older adults who show impaired pattern separation also show differences or changes in fMRI activity in the CA3 dentate hilar region. I think at the very least, these data indicate that integrating episodes into the context in which they're experienced is going to be more difficult for the older animal. Now, I want to mention one other difference in the impact of age on CA1 and CA3, and this has to do with awake behaving firing rates in CA1 and CA3 rats. So first of all, there is no change in firing rates during behavior in CA1. However, CA3 shows an increased level of activity or increased firing pattern. These are data from Ian Wilson. Now, I'm going to show you some unpublished data from young and old aged rhesus macaques that Alex Tomei and Daniel Gray are writing up right now. We recorded from baseline firing rates of cells in both the perirhinal cortex and CA3 region of the hippocampus. And what we saw was that there's no change in firing rates. These are box plots. The median firing rate are similar in perirhinal cortex in mature and old monkeys. But again, like the rat, I told you about the elevated CA3 firing, and like the increases in fMRI activity actually in older humans in the imaging studies, these old monkeys show increased firing rates in the CA3 region, which these box and whisker plots indicate. So the increased baseline firing rates also co-occur with decreased numbers of somatostatin positive GABAergic neurons in CA3 in the same animals from which these cells were recorded. Left is a section from somatostatin's GABAergic neuron in a middle-aged, and then right is the older monkey. There's some specificity to this, however. The numbers, there's no change in numbers of GABAergic neurons that express parvalbumin in this same tissue. So this suggests that the network dysfunction observed in CA3 region during aging in part arises from a loss of a specific category of interneuron, which we're really excited about following up. Okay. I want to show you now some more data from young and old monkeys that I've been working with out at the California National Primate Research Center in Davis. And these experiments focus mostly on behavior and imaging methods and continue this theme that cognitive dysfunction can emerge in hippocampal circuits without frank cell loss. Okay. David Amaral and Mike Bonacor have helped with the structural MRI scanning of 6 young and 6 old rhesus macaques. I mentioned earlier you multiply by 3 to get the equivalent human age, so these animals are equivalent to about between 27 to 87 human years. And Julia Chamy did standard volumetric analyses and found essentially no change in hippocampal volume across this wide age range from 9 to 29 years. In fact, it differed by less than 6%. So not surprisingly, there was no relationship between hippocampal volume and memory deficit. So in monkeys, at least, hippocampal shrinkage resulting from cell loss or other reasons can't account for memory impairments that we observed. And I think this is a really important observation because of all animals, it's only humans that show the classic Alzheimer's disease neuropathological pattern spontaneously. So we're looking at a living animal who, by definition, is Alzheimer's disease free, and it's simply just more difficult to be sure that you're testing humans that are aging normally. Okay. So these same animals, these same monkeys, were also imaged using an MRI technique that Scott Small helped us with that relies on measures of cerebral blood volume, or CBV, and is a correlate of basal or resting levels of metabolism. And because of the higher resolution of the method that Scott used, different hippocampal subregions could be measured independently, enabling us to compare activity across temporal lobe subregions in young and old monkeys. And again, in the same animals that showed no volumetric change, Scott found no significant change between the metabolic activity level and age in entorhinal cortex, CA1, or in subiculum, but a significant negative correlation between the amount of metabolic activity and age in the dentate gyrus. And importantly, there was also a region-selective significant correlation with the amount of metabolic activity and how well the animal performed on a recognition memory task. The higher the activity levels, the better the memory, suggesting that the dentate gyrus may be particularly vulnerable in normative aging. Now, we know that MRI measurements don't have single-cell resolution and are only an indirect measure of neuron activity. So we went to another method, and this method does have single-cell resolution and can reflect behaviorally driven activity in cells. This method monitors the activity of the immediate early gene ARC. And using this catfish method, which is capable of distinguishing multiple experiences in the same animals across wide regions of the brain at cellular resolution. John Guzowski, when he was on in my lab, first showed that the cellular compartmentalization of ARC mRNA varies as a function of time from the event or the behavior that activated it. So localization of ARC within the cell, so blue is the nucleus, so ARC is red, whether it's in the nucleus, like as shown here, or in the cytoplasm around the nucleus, can act as a temporal monitor of the activity or the behavior of neurons, either 5 minutes or 30 minutes before the animal is sacrificed. Now, besides using ARC as a circuit marker for cell activity, another reason to use ARC is that because we've previously shown that a knockdown of ARC in vivo, you lose late phase LTP, and also memory consolidation is disrupted. And remember, both LTP and spatial memory decay faster in older animals. So we thought that this would be a good marker to use in the aged brain. Now, to give you an idea of what happens with aging in the different hippocampal subregions, when you give animals 2 5-minute periods of behavior separated by 20 minutes, Monica Chawla in the lab showed that, that young, middle-aged, and old animals show the same proportion of ARC cells that have ARC expression. So the same proportions of cells, these pyramidal cells, show ARC expression in these regions. However, when she looked at the dentate gyrus, there's a significantly lower proportion of granule cells that are activated or engaged following behavior. So this suggests that, as in monkeys, the dentate gyrus in rats is a hippocampal subregion particularly susceptible to the effects of advancing age. Marcia Penner, when she was in the lab, had a whole huge series of experiments that I won't go through, but I just want to tell you that we think that transcriptional repression mechanisms involving methylation status of the ARC gene may play a role in preventing some granule cells from being activated. That is, we found a greater methylation in the promoter region of the ARC gene in the aged animals compared to the young, suggesting that these cells, the older cells, may be held in a state less conducive for facilitating ARC transcription. Now, these granule cell data from aging rats and the monkey activity marker data, along with some human data from Scott Small's lab, suggest that the hippocampal circuits that show the most distinct changes in normal aging involve the dentate gyrus hilar regions. I'm sorry. I've been talking too much today, I think. I've been having fun talking, but talking too much probably. So what I want to emphasize here, the point that I want to make here, is that this is a very different pattern. The pattern we find in normal aging is a very different pattern than we find in Alzheimer's disease. In Alzheimer's disease, it's not the granule cells, it's the CA1 pyramidal cells that are particularly vulnerable. Granule cells in Alzheimer's disease don't show changes until very, very late in the disease. So this at least favors the idea, I think, that while normative aging and Alzheimer's disease can co-occur, they have very independent patterns. So it's my contention, at least, that the disease is superimposed upon the aging process, not an obligatory part of aging. And way back when I first got into the field of aging, it was really thought that, you know, you're on a trajectory to degeneration and deterioration in that way. And I think this is pretty good evidence that it's not, we're not. What about other temporal lobe structures? So I want to tell you a little bit about the perirhinal cortex. This is just another example of circuit property changes in behaving animals, and I'll talk a little bit about these ensemble cell properties in perirhinal cortex in young and old rats. Now, we know from a lot of broad literature that the perirhinal cortex is important in perception and memory, and data supporting this are electrophysiological lesion and neural imaging data. And we've recently shown that in aging rats and aging monkeys, the older animals show reduced ability to discriminate between objects that show very similar features. Animals with perirhinal cortical lesions, these are data out of Tim Bussey's lab, show an exaggerated form of this object recognition deficit. Now I'm going to show you one of the perirhinal dependent tasks that we've given to our old monkeys, and partly to convince you that motivational variables are not a problem for these older animals. And this is one of my bonnet macaques, a 27-year-old bonnet macaque, doing this discrimination problem with a Lego task that I'll describe a little bit more for you in a minute. So she gets it right, and it's her favorite treats. And sometimes she gets it wrong. But anyway, so the animals, the older animals, definitely enjoy playing the game, just like the younger ones, but they are proportionally worse with more stimulus ambiguity. So I said that we used Legos so you can specifically and systematically manipulate between 60% overlap to 92% overlap. we saw the same thing in old monkeys as we saw in our old rats. This is the monkey example, that when there's more feature overlap and more requirement for pattern separation, the older monkeys perform worse. Overall accuracy is diminished in the older monkeys, and this is apparent, particularly apparent at the with the highest feature overlap in these animals. So both the rats and monkeys, the older animals show reduced ability to discriminate objects that share similar features. What about cell firing characteristics in perirhinal cortex? Sarah Burke did a comparison of ensemble firing properties in perirhinal cortex of young and old rats. She had many, many conditions in her study. I'm just going to focus on two. Two of the conditions that Sarah had was a no object condition in which the track was bare, so there was two food dishes, and the animal's job was to go around and it was separated by a barrier, so around and around to get food reward at the two dishes. And the novel object condition was 8 novel objects around the track that the animal had to pass by, going back and forth, and for food reward again at these 2 points. Now there were 3. These are this is data from the no objects condition, and the plots here, the red are the spikes, and the black the firing, the path of the rat around the track. And then these are the occupancy normalized firing rates. So there are 3 patterns of the no object conditions and cells firing in the no object conditions. First, a pattern where there's really no discrete, it's kind of mushy, not very discrete cell activity, or no firing at all, or a tendency to fire at the food dishes, which were the only actually objects on the track. Also, there was reward there. And this is an example of firing of object in the, this is firing of cells in a young rat in the object condition around the track. So these numbers, which you probably can't see, tell you where the location of the objects actually were. And this is the occupancy normalized firing rate pattern. And what you can see is that the firing tended to be clustered around the locations of the objects, and so we've called these object fields. And in this slide, you see 2 additional perirhinal cortical cells, but these were from older rats. Again, the red is the spikes, and the black the track around the path around the track, and then these are the occupancy normalized firing rate maps. What you can notice is that there is elevated firing around the objects as well in the older rats, but what was different in the old rats was the number of cells that actually expressed these object fields. So again, as for the hippocampus, in perirhinal cortex does not show cell loss with age. And there's a similar proportion of cells fire during rest periods or in the no objects condition between young and aged rats. But there is a decrease in the proportion of cells that show object fields in the older animals, either in the 20-minute or 2-hour delays between presentations of these objects. So this finding then of fewer cells in old animals could be a partial explanation for why older animals have a reduced ability to disambiguate shared features of objects, and it's a very clear finding in the older animals. So to summarize what I've just told you, older animals have a reduced ability to perform well on spatial memory and recognition memory tasks, and this happens in spite of no cell loss and healthy cell responses, but there are changes in functional synapses, plasticity mechanisms, and altered network properties. Okay, so I think as I mentioned to someone today when I was talking to them. While there are changes in perirhinal dependent and hippocampal dependent tasks during aging, actually the earliest, these changes happen during middle age and older ages. The earliest changes that happen actually happen in frontal cortical dependent tasks. And this rather depressing slide from Denise Park's lab shows changes in working memory in both visual, spatial, and verbal domains that are really, quite remarkably early. So together with Jean Alexander, we sought to look more broadly across the brain in the same monkeys that I showed you the structural imaging for. Remember, there's no volumetric change in the hippocampus. Jean first made a template so that he can do, for the monkeys, so he could do voxel-based morphometry. And what he found was voxels showing maximum negative association between age and gray matter volume were observed in the vicinities of the prefrontal cortex, especially left prefrontal and orbital frontal areas. That's blue to green. And overall, there was a difference between young and old age groups in the network pattern. The amount of network pattern. Now, what is the aging network pattern? That reflects the decline in gray matter volume seen across the frontal cortex. So the older animals had more of that network pattern. And more importantly, the better the spatial memory was, the less of this network pattern these animals showed. So these data suggest that reductions in gray matter in the frontal cortex reflect the effects of normal aging that can't be attributed to Alzheimer's disease. And I think a lesson to take home from this is that not only are the effects of aging selective within temporal lobe regions, there's going to be a wide variety of changes that you're going to see in different cortical regions with aging. As a further encouragement that animal models of normative aging may be relevant to humans, again, in collaboration with Jean Alexander and Ted Truard, we've used MRI to investigate the effect of aging on gray matter integrity in young and old rats. We used Ted's 7 Tesla system with 150 micron voxel resolution. And in a pilot study, there's only 10 animals here, but Jean was able to identify a regional network pattern that distinguished the young from the old rats. And this aging network pattern was characterized by reductions in bilateral regions of the frontal cortex, relative preservation of the thalamus compared to young rats. And the network pattern of aging was more distinct in the old group and correlated with a learning index score. So the more of the network aging pattern, the worse your behavior was in these animals. So at least there's some correspondence, again, across age. I want to include one other observation from the medial prefrontal cortex that involves looking at local field potential oscillations and single units. And this is from data that Nathan Insel collected in the lab that, where he observed slowing of the overall frequency of the gamma rhythm in the anterior cingulate cortex during behavior. Okay, so what Nathan did was to record decision and movement speeds on a 3-arm, 2-choice set shifting task. So the set shifts were between the light signaling where the reward was going to be and a tone signaling where the reward was going to be. And so once they got really good at one, then we changed the place in the stimulus, and that was the set shift. There were strong signals during behavior in the local field potentials during the task in a 40 to 70 hertz band. But what was significantly different between the young animals and the old animals was there was a peak frequency shift change from about 56 hertz in the young animals down to 53 hertz in the old animals. And this persisted across movement speeds and gamma period lengths. So I wanted to show you these data in part because while there's slowing of the gamma rhythm in the anterior cingulate cortex in these older animals, actually there's no slowing in CA1. So again, this change is very region specific. And importantly, Nathan also found a significant correlation between average decision time and the the shift in gamma frequency. That is, the older rats with slower gamma also made slower decisions, and this effect was independent of velocity. So in addition to age-related slowing in the gamma frequency and the local field potentials, virtually all neurons that were recorded were found to fire preferentially on a specific phase of the gamma rhythm as expected. But the slower gamma frequencies in the old rats were associated with a reduction by 1 to 2 milliseconds in the coupling between excitatory and inhibitory neuron pairs that were recorded. So that is, old rats showed a delay in the firing of fast spiking inhibitory neurons elicited by local excitatory signals either from regular or burst firing cells. And so we have thought about this for a while, and we think that because the firing of the fast spiking inhibitory neurons is known to be an important part of the control of neural synchronization within this gamma cycle, Nathan has suggested that this reduced excitatory inhibitory coupling may lead to this average lengthening of the cortical gamma cycle in old rats. And we do have a behavioral link here with slower decision times. So again, I want to emphasize the regional differences here. There's a volume decrease in the frontal cortex and gamma slowing. In the hippocampus, no volume decrease and no gamma slowing. And I'm going to summarize a little bit here so I can get to the last few minutes of the talk about the telemetry. So first, cognitive changes that seen with age. We see it in humans, monkeys, and primates, changes in spatial recognition and working memory. The neural substrates, well, the bottom line is it depends on where you look. For the hippocampus, there's minimal cell loss, no major volume change in normal aging, but the extent of connections, modifiability, and the emergent network properties do change. For perirhinal cortex, on the other hand, no cell loss again, but there's changes in this organized firing pattern around objects in old rats. So the network pattern is also altered in the perirhinal cortex. Now for frontal cortex, volume does change with aging, and there's slowing of gamma. And the last thing that I want to emphasize and let you go away and think about is that the pattern of normative aging is quite distinct from that in Alzheimer's disease. Hippocampal granule cells are particularly vulnerable in normal aging. In Alzheimer's disease, they target other hippocampal cell types. So for normal aging, a lot of people are thinking about good therapeutic targets being the granule cells in this Hyler CA3 region. So I'm going to go into acknowledgments now. All the many people who have helped with these various studies over the years that I've summarized for you here. If I do have a few more minutes, I'd like to tell you about some progress we've made trying to adapt telemetric recording methods for monkeys, since this may be relevant to some of you in the room. And I'll give you a very abbreviated version of this, I promise. Okay. So first, we set up a long run in the recording room. And we've trained animals to walk from this end of the run to the other end of the run for food reward. So you might be thinking to yourself, this sounds like something a rat electrophysiologist would try to do with their monkeys. Well, yeah, okay. So we've trained them to run back and forth for food reward. The tetrodes are chronically implanted in the hippocampus. They're movable. We can monitor the animal's behavior while he's running, while she's running in the room. And what we see is that if you look at the spectral density plots, we see an elevation predominantly in the alpha rhythm, 8 to 13 hertz, but also an elevation in low frequencies such as delta, about 4 hertz, and low gamma, about 35 hertz. This is blown up, so you can see some of these gamma bouts, increases in gamma bouts during walking in these monkeys. And Drew Moore presented these data at SFN this year, and he's convinced that, you know, when the monkey is actually walking, we're not getting what I might call theta, but we are getting something more like alpha. But it's not that we can't see theta. So there are these coupled theta alpha bursts. You don't see the nice theta that goes on forever in the rat hippocampus when the animal is walking. In fact, if you know, this sounds really weird, but the monkey theta is more bat-like than rat-like, because you get these small bat bursts of theta. You don't get this long continuous theta. What you're going to want to know, of course, is, well, are there place fields? Well, we don't know yet. We're still struggling with this, but I want to make sure that I get the acknowledgments to the recording technology development and telemetry experiments, and then I want to show you a movie. Okay. So these are, Matt's going to laugh because I think these were developed in the lab when he was there. So this is the rest condition, and she's nicely resting in her holding cage. She has her tetras in there. She's going to give you a nice yawn. Yep, she's nice and relaxed. And this is her in the running condition. This animal is 28 years old and still really, I think these older animals are really quite remarkable. So these balls are tracking balls from the Vicon system, so we can track multiple dimensions and so forth. But you see that she's coming down to get her yogurt. So it's Greek yogurt and strawberries that she's particularly fond of. And so that's what we give her, and you'll see her taking that. And then you'll see her face, which is full of yogurt, Greek yogurt, strawberries afterwards. But anyway, the animals are really quite, they like to play this game, walking up and down. And so if we have a chance to see theta or place cells or something, we think we're going to have it. But, you know, again, these things these issues, these kinds of experiments are long and hard. And certainly to get an age comparison, it'll be years. But, you know, I'm persistent as well. So thanks for being patient. Let me show you these better videos.
About circuit specificity in the hippocampus and changes with aging with the Dentate, Gyrus, and CA1. To what extent do you think that may be reflecting upstream changes like you showed with the perirhinal cortex?
Right, and I do think it's partly reflecting upstream changes. And so right now we have a big age comparison going on in the medial entorhinal cortex with a very elaborate maze system and that can change cues instantly and so forth. And we're trying to see whether there's any grid cell characteristics that are different while also recording in CA3 and CA1. So that's another study that will take years to figure out, but we're after that exact question. Yep. Bob?
So this is probably a naive question for the aging field. It must be debated all the time, but to what extent do you think these neural changes with aging are really specific to neurons, or rather than just general biological deterioration from microinfarcts and poor blood flow, some which could affect different regions differentially?
Absolutely.
Lots of peripheral factors.
Right. So there's not a lot of infarcts and so forth in rats, in old rats, and actually there's very little in old monkeys. I know Mark Moss, right down the road from you, has tried to make his monkeys eat this fatty diet that would, it's disgusting. He can't, change their he can't produce hypertension and all the bad things that he was hoping to produce. So these other animals are much more resistant to that. And I guess I do think that there's things like changes in glia, changes in the immune system that are contributing to these altered cell properties, but it can't be global deterioration because you see such specificity. If it was a global mechanism that was affecting most of the brain processes and so forth, you wouldn't see so much specificity and so much regularity between different individuals. So I think, yes, in human populations you have disease superimposed on normal aging. You don't have it as much in older other animals, and so that's about as hard an answer that I can give you.
So along that line, Tara, so what do you think is what's your speculation about the differential, deterioration of normal aging versus Alzheimer's? The CA1 more effective versus
Right.
anti-Gyrus?
I mean, so this really seems to implicate, the other entorhinal cortical projections that go to CA3. So the same cells in the entorhinal cortex project to CA1 and subiculum, but different cells in the entorhinal cortex project to CA3 in the dentate gyrus. So if the problem is CA3 in the dentate gyrus and normal aging, I would speculate that it may well be these other layer 3 cells that project to CA1 that are really impacted most in Alzheimer's disease, which is pretty interesting, actually. So that's why we're going to, in rat at least, we're going to medial entorhinal cortex first and layer 2 cells first to look at them, because that's what we predict is really going on in those animals. Back there, and then Matt.
So I have a question about the loss of somatostatin positive neurons in hippocampus that you observed. So how do you, so 2 parts to this. So the first part is, how do you resolve that with not a loss of overall neurons? So do you think it's being compensated by another neuronal subtype, or do you think it's loss of somatostatin within neurons themselves?
So I cannot answer the question of has there been a total survey of inhibitory interneurons in the aged monkey brain? No. So I don't know whether there's actually a loss of cells. So this could either be a phenotypic change, and the cells are still there, or it could be cell loss. But it's only of the somatostatin, not parvalbumin. And actually, I might have predicted it would have been the parvalbumin neurons that were altered, but they weren't.
That was actually my follow-up question was if you could speculate a little bit on what you think that means for aging.
Well, obviously it suggests therapeutic approaches that target those neurons, and it'll be interesting to see whether we can. If it's just a phenotypic change, then we have a target that maybe we can reverse and actually, turn those neurons back into functioning well. But I don't know. Yeah.
So how do you interpret your dentate results with respect to adult neurogenesis? So reduced fraction of cells could mean fixed numbers with increased, just more granule cells that aren't participating, or do you think of it as a reduced number relative to the available cells? And also
Right.
like the cerebral blood volume effects. How does neurogenesis, adult neurogenesis,
Fit into that?
impact those results?
Well, the numbers of cells, number one, let me just state that the numbers of cells happening and surviving for adult neurogenesis is relatively small compared to the millions and millions of granule cells. So it would be hard to detect a difference in absolute numbers if you counted them stereologically. And there has been there are no data to suggest that we lose granule cells as we age. So the ARC activity expression, the cells are there to count, but the ARC activity is blunted in these cells. So it's really the behavior being able to induce this ARC activity that is altered. We don't think it's a rampant granule cell loss. Now, neurogenesis does taper off, but it tapers off in middle age, and it doesn't go away. So after middle age, and some of these animals are, my adult animals would be considered middle age. So the comparison there is pretty even in terms of the amount of neurogenesis. But there's a lot to learn about granule cells, and I think they're, you know as well as I do, they are so difficult to record in the freely behaving animal because they're so quiet. So few of them ever fire, and so if you're lucky enough to have your electrode in there, that would be great. And that's why we like these activity markers, because we know they're out there, but it's a really small proportion of them. So they're an enigma, and I think that we have the opportunity to improve memory, possibly through more neurogenesis and good cell function. But I don't know why that should work when there's so few neurons that actually do survive, and why they would wire up correctly and all of that. It's just, but the jury is out, and there's some pretty striking data that suggests that when you manipulate neurogenesis, you also improve memory. So.
Well, if no more questions, thank you, Carol.
Thank you.
Thank you.